because
They're
they're
there.
like 28.
Underwater, and they're not going to bite you.
They're 2.8 kilometers below my feet right now.
How
This one might be a swimmer.
Oh.
Oh, it's taken off again.
Oh.
Ooh,
Hello. That one's going zooming in. Oh my gosh. Whoa.
Look at it go.
It's really mesmerizing.
It is.
Isn't it?
Look at it. Oh my gosh.
Polykit, go.
Yeah. Yeah, so
Wow.
this one's called swimmer.
This is so cool.
Oh,
Oh
wow.
my
Look at
Gosh.
that.
Oh, this footage is incredible.
Oh gosh, hard to keep in focus.
This is incredible. Oh my gosh. Wow. That is gorgeous.
And it has these special little palps, those little kind of antennae-like projections that they can drop. And so their common name is like green bomber.
Do you know of any aquariums that keep polychaetes? Because these are adorable.
Usually they just like have polychaetes for using as feeds.
Really?
They don't usually keep them as like things that people can observe.
Oh, I could look at this
Like
all day.
there might be in the aquarium, but these animals are really small, and so you wouldn't see them. And many times polychaetes are burrowers, and there are only a few of them that swim like this. Well, not a few of them. There's a lot of them that swim like this, but not many that you would see in an aquarium.
Wow.
Yeah, they usually are considered aquarium pests.
I think again, tugged a bit.
Yeah.
Oh, that footage was incredible, though.
Was
oh my
cool.
gosh.
You kind of went off on a little chase there. Yeah, back to the plan. My alone, turn. There we go.
Noting quite a bit of collapsed ferromanganese crust in this area. Not quite sure why yet. There's also a substantial amount of sediment below it. Doesn't necessarily mean that the sediment is older, though. Just means that the sediment found a place to settle down below it and build up. But, that could be structural. It could have something to say about the substrate. Don't know yet.
Is that something over there? Like a... Oh, wait, no. Are those my eyes playing tricks? Is that a fish over there? In the corner?
Yeah, it looks like there might be something there.
No, it was like further.
Is that what you were spotting?
No, it was like out of frame. It went out of frame.
Ah, okay.
Looked like a dark figure in the distance.
A dark figure in the distance.
Yeah, that's another sponge stock.
I'm kind of loving this terrain, though. It looks so peaceful.
Oh. Yeah, sometimes it just like says, hello, I'd like you to look at my pop-up.
So are we aware of sea pens occurring at these depths?
Yep, there will be sea pens here.
Okay, if the sediments are thick enough, right?
Yes.
Yeah.
Probably Umbellula or Cophobelemnon or Funiculina.
Okay.
And, you know, we could also see rock pens.
Mm-hmm.
But, yeah, I don't see any here.
No. You're reading exactly why I was asking.
Now, I do scan the sediments because sometimes you see sea pens or anemones or even cup corals
Yeah.
that can root in sediment. There's sponges, too, that'll root in sediment. But the cool thing about the sediment is if we took a big scoop of it, there'd probably be a bunch of stuff in the sediment
True.
that we're just not seeing because it's not on the top.
Just more questions.
I'm not confident that it's
I think there was
thick enough here for a sediment
coral
sample.
over to the left of this from the Bersingid.
Okay. Oh, yeah, good spot. Another little bamboo, right? Oh,
Probably.
there it is.
Oh, wow.
Yep, tiny bamboo.
Yeah, I'm eyeballing
That's
the
the same
sediments because we did get a request from shore team for some sediment, if possible, but I don't see anything that's thick enough around here to get a good scoop with one of the cores.
No, yeah. That's really hard to do on seamounts.
It is.
They're usually more rock than sediment.
Yeah, and it's really hard to get them to retain, so I've really struggled on these dives to get a good sediment core.
Yeah, the sediments are coarser and they're not sticky. They don't stick in the core tube.
Good.
Yep.
Yeah, I can see at some of the rocks I pull up where you do get like carbonate mud, but there are also a number of instances where I've been getting some marine sandstones coming up as cobbles, and those are ranging anywhere in grain size from you know, like kind of a fine sand or a large silt size all the way down to mud. And yeah, those larger grain sizes, I think you're right, those are what makes it problematic. So it's something to keep an eye out for, and maybe we'll give it a shot later. But
oh, definitely not here. It's
yeah,
definitely
this isn't thick enough where we'll be able to pull something off, unfortunately.
Yeah.
We are looking though.
It's so easy on the abyssal plain. It just like
It's just all sediment.
yeah, and it's so sticky and fine.
Isn't that how science works, though?
Yep.
They always
Yeah.
want dirt where there's rocks and rocks where there's dirt.
You can't ever please us 100%.
Although I have been told by a marine technician that the two happiest people on research ships tend to be also the messiest when it comes to sample retrieval. at least on like some of the like really deep sea, like more focused cruises. You get the geologists who just like end up wearing ferromanganese. And then there are the mud people who do like mud coring, the mud geologists.
Ooh,
and
what?
they sample that stuff on deck, and just apparently just the entire ship gets covered in mud, and they cannot be happier about a good mud sample. I'm serious.
That is so
Like,
cute, though.
So what you're saying is geologists are the happiest.
Oh.
So you've listed 2 geologists.
Well, those are more geology-focused expeditions, so yes.
I love that.
I think it was the day that I got really covered in ferromanganese last summer after a particularly messy dredge,
Yeah.
and she's just like, "It's you, it's you guys and the mud people, I swear."
You guys and the mud people. Oh my gosh. I I love that so much. I would love to go to geology conference one day and just hear the stories.
There's some good ones. You kind of have to get to know some people though.
Yeah. That's awesome.
I see the excitement expressed in biology in a very different way, you know. Also, they tend not to end up inadvertently wearing their sample as often.
Yeah, generally not.
There are exceptions to that. I mean, I've heard some of the whale stories, but
yeah, well, like,
yeah.
we don't want to be wearing that.
Yeah.
No, you don't.
Not
It's
for
not
fun.
good. Been there, done that.
I know
Had
you-
to throw away the t-shirt.
Oh my gosh, no. Oh no.
But no, there's always palpable excitement in the lab when there's something.
Well, it was a whale that got struck by a ship, and we pulled it ashore and did a necropsy.
Oh.
Uh-huh.
Oh wow.
Yeah. It is pretty palpable in the lab though, and there's a new species being documented, or it's been something that's been long on somebody's wish list.
Yeah.
Yeah.
Oh. I
Dead
love that.
whale is the worst.
It is the worst. It's so bad. The smell just sticks to everything.
Yeah.
Really? Oh,
Yeah.
is that a fish?
We've sampled
The top corner?
whale falls
The
before.
right?
Yeah. Oh. The older it is, the worse it is.
I think I missed it.
Oh, dang. It looked like a fish. Swimming around.
They had some whale bones
there it is.
that they wanted to
There's
make
a
an
whale.
artificial
In
whale
the
fall
bottom
with.
corner. It kind of
In the minus 80
Dang.
freezer on the KM. And then during COVID, they were in shipyard.
Oh, it went down?
And yeah,
Just
they
throw it away.
yeah, no, they had to throw it away.
Oh my gosh. Whoever's collecting the trash. Oof.
It was bad. It was horrendous. Everybody was
what
not
is
having
this?
bamboo.
Big bamboo.
It's huge.
Bigger than the ones we've seen.
Look
Yeah.
at that. Oh,
But
that's
it looks
incredible.
pretty similar otherwise, morphologically.
This one might be different. I'm looking
Oh?
at the way the polyps are arranged on it.
Okay.
I'm gonna tilt up. Yeah.
Whoa.
Oh,
see.
okay.
It looks like they're all, yeah.
Wow.
These are
this is
smaller.
different.
Yeah, you're
This
right.
one is Bathygorgia.
Okay. Yeah, this has much smaller
So
polyps.
all
The other one had
polyps
monster
are on
ones.
one side, and the other side is smooth, and it does have the smaller polyps.
Wow.
Am I seeing some orange associates on this?
Yeah, those
Oh,
are little
yeah.
polychaetes
Okay.
Oh,
that are there. I'm
wow.
gonna. Ooh.
Are the polyps more dense on this one, or is that
Yeah,
just...
it is more.
think
It's more
so.
dense and concentrated on the one side.
Yeah. Could that be due to like ocean currents or?
No, it's a species specific.
Oh, it's just
genus
how it does
specific.
that. Oh, wow.
Because it's actually all the polyps are on the side opposite the current, or
Mhm.
the current.
Right, and it can kind of twist and move, but
Oh.
yeah, generally the polyps will be oriented down from the current so that as the water passes across the fan or the colony, it'll cause turbulent flow, and they can have better chance of capturing particles.
Oh,
If you're
how
facing
cool is
in,
that?
you would just like kind of have to like, gotta catch it on the fly.
Whoa.
Dude, that's awesome.
Yeah. Got fluid dynamics. That's
They
cool.
do wind tunnel testing with it?
Yeah,
They should.
Go to wind tunnel, test your
Put it in the
corals.
tunnel.
Honestly, there probably has been done with the coral to sort of see how that feeding mechanism works. Like you
Put
you
some
can
dye
dye,
out there,
yeah,
yeah.
you put some dye and you watch it go past and how
Yeah.
the colonies will, or how the polyps will grab little particles and things.
Wow.
I'm gonna go look at the base.
Okay. Is there any interest in a collection on this, or are we gonna let it be?
It could be a good potential collection.
Okay.
Though we do have 24 hours on this dive, and there might be good stuff upslope.
Right.
So I'm
yes.
not sure.
We already filled up three buckets. So.
Yeah.
Yeah.
This has to be,
Do you have any thoughts, Louisa?
yeah, Louisa.
No.
You're the most knowledgeable on the bamboos.
No, no.
Of all of us here in this room.
I'm the most frustrated with bamboo.
Oh no, I can imagine.
Right,
we'll let it go.
It's got
Yeah,
a little.
let's let it go.
Thanks for making the
Okay.
choice for us, Robert. It's like,
The pragmatism of the pilot. Well, we did get some excellent footage regardless.
yeah.
Little shrimp friend.
Just mixing it up.
Wow.
It does feel like a 305 kind of move, doesn't it?
Right. I was like, just gonna. That heads us straight toward Waypoint 7 and kind of like forgetting about 6, but
It's all good.
yeah.
I mean, there's
It
suggestions.
goes more perpendicular to the
Are you
contours.
interested in this at all?
Yeah, let's look at this thing. It looks kind of dead.
A little bit.
Yeah, it's definitely
Very
dead.
Dense holes, very high contrast. So yeah, that netting
Yeah.
that you often see growing over them doesn't seem to be there.
But, you know, it was a glass sponge. It is no longer. It is just a glass framework of
It's
X
a very
sponge.
pretty one.
It is.
Sort of like that dead parrot skit, you know.
I think we had someone in the public chat asking earlier about where the silica comes from to construct the skeletons.
Well, it has to come from the water.
That must be some very slow growth because water does not typically have readily
There's
carry a
another
lot of
sponge up there.
dissolved silica.
That one's a live sponge.
Ooh. That one looks so neat. Look
Well,
at that.
how else would they get it?
Exactly, yeah. I mean, they're not pulling it out of the rocks
Well,
because
they move a lot of water though, right?
They do move a
Oh,
lot of
They
water.
do, yeah.
That's all they do.
Wow.
So it doesn't have to be like heavy concentrate to get stuff
Yeah,
out of it.
just have to filter enough water.
Is that why they're kind of slow growing then? Is
Mhm.
that reason? Wow.
That is a factor.
Wow.
Food and resources and nutrients.
That's looking a little yellow there.
Yep, this is
Yeah.
one of those yellow bolosoma.
Yeah,
That's
I
gorgeous.
know you like the yellow bolosoma.
they're cool.
This one isn't as yellow as some, but
It's yellow, it
it's
counts.
yellow.
Yeah.
We call this bolosoma bolosoma species B, because we don't have a name for it yet, but it's species B.
Is it being characterized right now?
I know there are samples of it, but I don't
Okay.
know if anybody's working on it.
Interesting.
Gotta love a good bolosoma.
Sure do.
Yeah, it's gorgeous.
The affectionately known by some of us as cheese moon wheels.
Oh, the
So,
cheese
some of them develop heads on the stalks that are very circular.
Oh,
and when you look
really?
at, especially some of the bigger ones, and when you look at them face on, they look sort of like a slightly yellowish moon,
I love that.
or like a wheel of cheese with some extra big holes in it.
Every time I see wheels of cheese, I just think of Wallace and Gromit.
Oh, that
Look
is
at
a
all
sediment
these.
field. Look at all those basalt pebbles.
Wow.
It's a field of them.
Sure is.
It's just so neat how fast the terrain changes as we go. It's crazy.
So this is where things are widening out laterally for us. We lose a distinct ridge for a little while, and we'll pick it back up higher up. So this is some sort of little morphological terrace that we're on. So this is collecting a lot of sediment and a lot of rock pebbles from further uphill.
Wow. What kind of like, if it's coming from the hills, oh.
All right, what was the question? Sorry.
Oh, no, you're good. So for something like this to form, is it caused strictly by the incline of this, or? I don't know, I'm just trying to understand.
So this
how
is
it's
like a
formed.
sedimentary fan in some ways,
Yeah.
where this is just an area that's flat enough that and extensive enough. So if you see like over here, how you have this kind of wide area here, and then that slope's really picking back up. We're seeing some of the most like distant sedimentation processes from that. From that uphill process. So this is where the stuff that managed to travel out the furthest because it's the smallest
If
grain
you could
size or
suction
whatever,
this fish for me, I will be so
managed to
happy.
come out this way.
Yeah.
Yeah.
Are we going to attempt to suction?
I mean, we've got a lot of bottom time.
It's a high challenge. What kind of fish is this?
This is a rattail fish.
Oh, it's a rattail. It's so fast.
I don't know if we'll be able to get it, but
Oh,
no,
oh my gosh. Oh my god.
Oh, we got a good look at it.
we did. Oh, there it is.
Hello. Goodbye again.
Yeah, there's no way. This one's a spicy one.
It is
Spicy.
Spicy.
No, Levi.
It wanted to be collected, apparently.
It's like go into the suction.
Swim right through Herc.
Yeah.
There it is.
Oh.
It's right off the
porch.
port.
Oh.
Yeah.
Yeah. Oh, cool. That was a nice spot.
There we go. At least we got one rat tail. It wasn't Coryphonoides. It's probably, it looked like Kumba. A cute little one. So adorable. I just want to see a nice big rat tail.
Yeah.
We are likelier to see some fish in the sedimented area.
Yes.
Tend to see more in these places.
Oh.
So this is a good place to go fish hunting. Or at least fish spotting.
Because that's where the fishes are hunting.
Yes.
Oh.
All sorts of critters burrowed in the sediments.
Yeah, you can find lots of tasty treats around here. That's my favorite thing about doing the drop cameras is you just see who gets to come and like when they arrive. And so you'll see the shrimps are usually one of the first ones on the scene, and the rat tails tend to be one of the first ones on the scene. eels will come, and you'll have your slower moving, more more ambulatory friends coming later, like the lithodid crabs and the urchins.
Yeah.
They're not as quick
That makes
on
sense.
their feet because they're not swimming, they're walking. But it gets so exciting because every video you're like, what am I going to see now?
Do you get a lot of sea stars on the drop cams too, coming in after?
Not usually the sea stars.
Okay.
Yeah. The pancake urchins come, the big echinothurids.
Yeah.
They're usually the last to arrive.
Okay. So they just kind of clean up the last bits.
Yeah.
Gotcha.
Well, they don't have to clean anything up because the bait is contained in a container. The shrimps are really the only ones who can usually get to it because they can stick their little hands through the holes.
I see. Okay.
At the shallower deployments, at 250, 300 meters, the heterocarpus shrimps usually clean off all of the fish, and all you have that comes back are bones.
Oh.
How long does that stripping process usually take?
Oh, well, we have left them down for about 14 hours, so within 14 hours.
Wow.
Whoa.
That was a lot faster than I was expecting. I was thinking
Yeah,
like
well,
days to
this is
weeks.
shallow. When I say shallow, it's
True.
relative, because like 250, 300 meters seems kind of deep to some people, but yeah, they move fast. But there are some deployments where the amount of shrimps that come, you're not surprised that there's nothing left,
Okay.
because it's like so many shrimp. It's hard to count them.
That's wild.
And there was one deployment we did on a seamount in the Clarion-Clipperton zone where there was hundreds of Synaphobranchid eels that came.
Whoa.
It was insane. We've never seen such a high number of them at any baited camera. Like, it was really, really impressive.
Wow.
That's a really interesting place. There's a lot of research going on in the Clarion-Clipperton fracture zone, because they've also been doing some of the experimental, like, ferromanganese nodule, like, just seeing what the seabed looks like and how it recovers after stripping the nodules away, too. So not just, like, a lot of biological work, but there's just so much happening in that area, scientifically.
And
Wow,
we're
it's
discovering new
wild.
species all the time over there. It's such a unique environment.
Uh-huh.
Yeah.
And the nodules themselves are a very unique substrate.
Yeah.
There's little primnoid corals that grow only on the nodules.
So that's a really unique environment to maintain.
And like you said, like the nodules are just precipitated ferromanganese that takes millions and millions of years to grow.
Yeah.
So disturbing that, it's never going to come back, basically.
Not within time scales perceptible to humans.
Oh, absolutely not.
Yeah.
There's that little floaty thing. Yeah, right middle below the lasers. Yeah, that. What is that? It's a sponge.
Oh. That one looks interesting. It's like...
Oh. So this is one of those sediment rooting sponges called Hyalonematidae.
Does it have an associate on it?
I was looking at that.
Yeah.
I'm not sure if that's just, doesn't look like it's alive, whatever that little string is.
Yeah, that's the thing I was confused about too.
Oh, I know what it is. It's an amphipod tube. So we'll wait for it to bounce back. You might see the little amphipod on the end. You see that?
Uh-huh.
So the
over here?
amphipod's like right underneath the bell of the sponge. When it
Okay.
bounces back up, you'll see it.
Oh, yeah, I think I saw it peek out there.
So the amphipods actually make these little tubes, and they like to hang out on them.
Wow.
And I've never seen them make one on a sponge before.
Oh, that's interesting.
Yeah,
Yeah.
it was neat. Normally when you're hanging out on a sponge, you just hang out on the sponge, but it made a tube on the sponge.
It made a pretty impressive tube.
Right? Yeah. And how deep do these sponges usually root?
It's not too deep in the sediment that the roots go.
Mm-hmm.
So the roots are just basically really long spicules, or it's like a long piece of fiberglass.
Oh.
I wonder if you could use them like as fiber and send little fiber optic messages down the line.
Oh, that would be cool.
I mean, theoretically,
Right.
it's the same thing. I don't know how pure it is, and how well it would conduct light, but it would work technically.
Sponge communications.
Right? I'm pretty sure that's not what they're doing with it.
Yeah. What does sponge have to say? Filtering.
I'll bet they have a few things to say.
All right, we should probably get going.
Yeah. All right, I've been pinged by the shore team that this is the kind of substrate where we might find abyssopathes.
Yes, yes. They also root in sediment, which is kind of unique for a black coral.
That is a sea pen.
Umbellula.
Oh,
See,
question
you asked
answered.
me whether or not we would see them, and yes,
Yep.
yes they
Wow.
are, right here.
Yeah.
Oh, that's
I love it when nature
gorgeous.
does that.
Is the dark black areas, is that the skeleton that we're seeing?
Oh,
there, or
no,
what
that's
is
the
that?
stomach.
Oh, it's the stomach. Oh my
Yeah.
gosh.
Yeah, see
Oh,
how
how
it touches to the
cool.
center, the, like mouth, and
Whoa.
so that's the gut of the animal.
Wow. So for like the gut, is it like everything goes in and out the same way, or does it go? in the
No, they
center?
have an anus as
Yeah.
well.
Wow.
So it's like a U-shaped situation.
Oh. What an interesting gut.
Is that a small tunicate behind the Umbellula on the left? So right here?
It might be a small sponge,
Okay.
actually. I think. Yeah, let's go.
Moving on.
I called in a large move so that we would see stuff. Because we only see things when I put a move in.
That's how it always goes, I swear. Yeah, if it's translating down the cable, we gotta fly.
Yeah.
I'm gonna remember to wear my longer socks for the morning watch.
Oh.
Yeah.
I'm wearing
Ankle
ankle
surf.
socks, and my ankles are getting quite cold now that the AC is cycling again. It's okay though, I'm fine.
Let's find some black corals.
Yeah.
Trying to manifest that.
Manifesting.
Sometimes it works.
Yeah, I believe it.
Oh,
shrimp.
yeah, that was Actinoscyphia.
A little shrimp.
I can tell because of the way it was swimming.
Oh.
Gotcha.
How is it swimming that was different?
Sideways.
Wait, is the name Acantha what?
Actinoscyphia?
Thyridae. I thought it was Acantha sideways.
Actinoscyphia, yes, we should call it that.
That would be
Chris
very
Kelly would call
appropriate.
it the drunken shrimp because they rarely swim in a straight line.
Oh. Does anybody know why that is and why, like, is it advantageous or?
I don't know why.
Yeah.
I think that's how they do.
Drunken trip, do as they do.
You know.
Yeah, sometimes behaviors or features an animal, aspects of an animal like that don't necessarily evolve either because of like an advantage or a disadvantage. Sometimes it's just, there is no advantage or disadvantage, so it doesn't necessarily get, there isn't necessarily pressure for it to be in or out of the genetic pool.
Yeah.
Yeah.
Yeah, they're just kind of floating around here. And they can control which direction they go. It's just,
That's
most
important.
of the time, it doesn't really matter.
Yeah.
Petting tug.
I see a shadow on the sea floor. I'm not sure.
I'm getting pulled.
Yeah, that sort of trident-looking shape.
Yeah.
Oh yeah, right by the lasers.
It has potential, but it's nothing.
Oh, sponge stock, partially sedimented.
We've been fooled.
That looks much different up at elevation.
It
Right?
did.
Yeah, it tricked us all.
I was like, oh, that could almost be a black coral.
Right?
Almost.
And I'm just over here like, at least that's not a sea star / sediment.
I'm just like, has potential.
Yeah, no, I get it.
Yeah. Well, she's coming. You just gotta wait.
Something here.
Oh, nice spot.
Ooh. It's very
sea
bright.
star.
Oh.
A very out of place Brisingid.
Yeah.
It's not a Brisingid.
Oh, it's not.
This
Oh,
is an
yeah.
Ophiocanthid brittle star.
Oh.
I think earlier I was telling you that the disc looks a little bit like a hibiscus flower.
Yes.
Yeah.
Oh yeah. It
Now you can really see that.
Yeah,
But
you
I
can
can
see a
see
leg
why you
regrowing
would
here.
think it could be a bracingid. It has a similar look.
Like the spine, like the orientation of the spines
Yeah.
is very similar.
Wow.
So is this generally solitary? Like it's not typically found as an associate on anything?
Oh, they're usually always associates. This one is on a walkabout or something.
Ah, so
Take
So it is out of
in.
place.
Yeah, these are the ones that we were seeing on all the sponge stalks, and we see them on those yellow bolosomas.
Yeah, and when
they-
you mention the scientific name, I'm like, that is usually a name I hear with corals or
Yeah.
sponges.
They're usually on other things.
So it is
this one
very
is
out
not.
of place.
I mean, they can be on the ground, but they usually don't
Yeah.
do this.
Yeah,
on
it's
average.
also been through some things because I see parts of some legs missing and
Yeah,
regrowing.
maybe it got displaced.
That's what I'm wondering, yeah.
Oh, I didn't think of that.
Yeah, I've seen them fall off of their, you know, whoever's hosting them multiple times, but...
Oh, yeah.
These are the dramas, right? Where they're
Yeah, yeah.
Oh, no.
This is the first one I've seen one trekking across the plains.
Have you ever seen, like, brittle stars move? Like, they can really move when they want to.
Really?
There's some
Oh.
wild
Did you
time-lapse
participate
videos
in
of those.
2017 Okeanos?
Me? No.
No. There was one dive where the seafloor had a whole bunch of brittle stars on it, and there was this really adorable bobtail squid, and we were all oohing and awing over it, and it got a little too close to the brittle stars, and the brittle stars attacked and took it down. It was so devastating.
No way.
Oh my gosh,
Yeah, it was
what?
wild.
Is that something?
That is something.
Oh,
Don't ask me what it
I had
is,
never
though.
seen anything like that before.
that's crazy.
Man, that's brutal.
Yeah.
What is this?
This is a Homerian asper, the blind lobster, and it's partially buried in the sediment.
Oh,
You don't see that very often.
how
Yeah.
precious.
Unless it's dead, then
Oh, that would
Maybe
be.
it's just sleeping.
maybe it's just
It's
sleeping.
just resting.
Resting my eyes.
Yeah.
I don't know. Only one way to find out. Poke it.
Okay. Ooh. Slurp it. Beautiful
Okay,
color,
fine. I'll
though.
go get the pool noodle.
Poke, poke. Wow.
Usually we see those sitting out on the rocks.
Yeah.
I don't think I've ever seen one buried in the sediment like that. It's kind of
Me either.
strange.
Yeah.
Maybe because they don't have anywhere to hide, really, maybe? Or,
I don't know.
yeah.
Oh,
There's
okay.
a new thing we haven't seen on this
What
dive.
is this?
Yeah.
This is an Actinoscyphia. It's a type of anemone that tends to always grow on stalks. So it grows on, this is an old sponge stalk, and as we zoom in, you can kind of see how the spicules, the fibers, are all twisted together.
Yeah.
And that's what the inside of Herc's tether looks like.
Oh.
It doesn't look anything like that.
So what you're saying is this is how we get fiber optic cables.
Right. We harvest them from the deep sea. Not the other way around. And
They're
there's also
naturally
a cool
growing
little
one.
isopod that's hanging
Sitting
out.
in it, right?
The little white thing, yeah, that's a little isopod,
Oh.
which is related to pill bugs or roly-polies.
Oh.
Sure.
I love roly-polies. What a beautiful anemone.
I really like isopods.
They're precious.
but my favorite is the Bathynomus. That's a giant isopod.
Yes, that one's so cute.
I have a larger than life-size plush.
Oh, what? I'm so
Yeah,
jealous.
it's like slightly larger than life-size.
Oh my gosh, I love that. Oh.
I like to take it. I like to take it to sea with me, but it didn't really fit in my bag this time.
Oh.
I did bring my shark though,
Oh.
my IKEA shark.
Oh, I love the IKEA shark.
Yeah.
I have the IKEA bear.
Oh, and there's a little scale worm on here too.
Oh, yeah, look
See
at
the
that.
dark purple, and then the white is the isopod.
Yeah, so there's the isopod, right? Yeah.
Yep.
And then, is that the other one, or is that part of the anemone?
That's the scale worm,
Scale worm, got it,
the
got it.
polynoid.
Polynoid, okay.
Wow.
Cool, cool, cool. Probably should get going.
Yep. Sounds like one of Next Watch's members may have just come in.
Yeah, it's about
It's
about
getting
that
about
time.
that time.
It is about
that last
that
hour
time.
flew on us.
It did.
I know. Lots of good stuff.
Lots of good stuff.
You know, my favorite thing about the deep dives is that there's really good pacing and you get to look at everything instead of like the really dense dive, which was gorgeous and I'm not complaining. It's just
It's so
we
much
don't get
harder
to look
manage.
at everything because you feel so overwhelmed and you don't even know what to look at.
And there's a lot more to manage there.
Yeah.
As far as dive details. Because, yeah, it's like balance. You have to balance, okay, what do we need to do here? What do we need to look at? But also how far, you know, how much time can we spend here before we need to move on?
Yeah.
And that's a lot of difficult decisions to make.
Yeah.
There's another Umbellula up front,
Ooh.
looks like, in cinema camp.
Oh, yeah.
Oh, okay. Oh, yeah. There it is.
That's an Umbellula.
Yeah. I like those.
Wow.
Good.
Roger. What you got me already?
Okay.
All right, so we are at waypoint 6, and we are starting to move toward waypoint 7. So
On the move.
watch change happening right as the slope begins to pick up again. And hey, there's the slope.
There it is.
Hello.
Well,
Perfect transition.
Hello.
part of it locally anyway.
That's something on the rock. See something kind of
Yeah,
red
we're suddenly back into
color.
rock territory. So we will probably move out of sea pen territory and back into more hard substrate animals.
Do you need a
Potentially some
closer
more
look at the
corals.
star, or keep moving?
I think keep moving. I think that's a percentage.
All right.
Video watch change.
Alrighty, y'all. 8 to 12 is currently in the process of signing off. Thank y'all for tuning in, listening to us, but stay tuned. More exploring to come. This is a 24-hour dive, so we'll be on for quite a while.
Yeah, you will hear from us again in the morning.
Looking forward to it.
All right.
How you doing, Nav?
Nav is alive.
What was the bearing of the last move? Or I guess what's the bearing to waypoint 7?
Let's see. Bearing 305.
Roger. All right, so let's bring Atalanta to starboard.
Hi everyone, we're just going through our watch change. We'll be right back.
Megan says she's been taking bigger steps, but so I guess this incline isn't too bad.
Yeah.
We'll see. We'll
Okay.
get a-
Yeah, the Atalanta sonar looks pretty soft.
yeah.
Roger. I'll keep coming over. Shrimpies, a bunch of rocks. These rocks.
Multipuffer's having an issue.
Oh, video, you there?
A yellow one? Where'd that paper go?
Go for video.
Oh, the jelly? Go for
Oh,
jelly?
jelly.
He took it.
Oh, yeah. How do you feel about suctioning that jelly?
Can you guys, I need to get the power recycled on the multipuffer.
All right, zoom out.
Zoom out. How do you feel about suctioning that
Yeah, I'm
jelly?
going to drive it to the nozzle.
Okay.
Why don't you give me to a jar?
Are we sampling him? Sampling it?
We're going to try,
All right.
since we've got lots of buckets available.
Maybe can you
Jar 4.
tilt the camera down to the suction nozzle,
Gonna
please?
try
Keep
and
the jelly in view.
switch.
the jelly?
Right by lasers there. So down right.
yeah,
So
I
tilt
see it.
camera down right.
Let's play a fun game called How Fast Can I
Alright,
Log
and
into
next,
C-Log?
What jar should we go to, Steve?
Say again?
Which jar?
4.
Hurry, Leela, hurry.
All right, let's index to jar 4.
Yeah, sorry, I'm trying to find it on my freaking. Hey Data, what would be the next?
Do you think,
This is going to be 251.
do you think you could do one more zoom, or are you situated?
You gotta get yourself to a jar.
I'm moving to jar 4 here. There we go, that's 3.
Yeah.
That's 4.
Oops. Oh, we have somehow the same computer.
You just driving it.
You ready? You want to start suction?
You gotta do it on. I haven't.
I'll do it.
I'm just going to drive the
Go
nozzle
ahead.
to it.
Oh, let's swap. So I'm going to take
Yeah, start
chief
suction.
side, and then you're going to take side.
4.
There you go.
Yeah. Start it. Yeah.
Suction on.
I don't see it. Stop it.
There
Okay,
you
start
go.
it back
right there.
up. Is it going?
Yep, it's on.
All right. Where is that thing? It's below me.
Yeah, down to the right.
You're lined up.
Right in front of the sucker.
If you guys can tell
Ooh,
where
ooh,
it is.
get in there. Oh, it's in. You got it.
Okay,
Nice.
let's suction on 3, 2.
Did it make it?
Yep.
All right.
It went in the hose. I don't see it in the jar yet.
I don't see it in the jar either.
Turn it back on. There you go.
There it is. Yep. Yep.
Yeah.
Okay.
Hi,
Nice.
can I ask you to turn the power off to the multipuffer?
Yeah.
We need to recycle it.
Yeah.
Okay, so that's jar
Jellyfish
4.
or
All
nidarians?
right, power's off.
Yes.
And
Yeah.
Thank you. Leela, that last sample
it-
that was going didn't actually go very far.
Okay.
So, the pump seemed to have stopped a while ago. So,
Okay, I'm noting that down
yeah.
in 1 moment.
I'm definitely awake now.
I know.
That was everyone's wake-up call.
We're in it.
My heart is beating so fast right now.
All right, let's turn it back on.
And we're just getting started.
You need a closer look at the sponge or keep going?
We can take a snap on the sponge.
So
So
snap it video.
copy. Jellies are kind of a snap judgment, but we do have a request in our expedition report for gelatinous plankton. And there is a team of scientists ashore who is very much interested in all of the rare jellies we might get. And usually most of them are rare.
Did we get the power back?
No, you want it back up?
Yeah, please.
Fixed video, we can zoom out.
Power's back
Copy
on.
going
Thank
wide.
you.
Good snap.
Just realized I've called it jelly instead of jellyfish in everything I've done thus far.
Jelly is, yeah, probably the better term than jellyfish. It's actually, it's probably a,
Leela, I'm aiming to start pump 7 right now. Let's see.
Are we skipping pump 6? Oh, because it hasn't been working?
Yeah, 6 hasn't been working.
All right, let me know, is pump 5 just ended now?
Yeah, so pump 5, okay, so pump 7, let's just get this. Pump 7 just started. It is working now.
Okay. I would prefer to submit the end for 5 first, if possible, but if you think it's still going.
Oh, I'm just right. What do you think? What's the depth? I just want to get the depth.
2763 meters.
Okay. And so pump 5. Hang on, I took a picture.
You can probably do 20 meters.
Is it, well, I don't need the mills quite yet, but is it stopped?
305 is fine.
Yeah, it stopped. Okay. Apparently it stopped a while back.
Okay.
So it didn't, it probably stopped a couple minutes after it was started.
305.
Fish there?
Nav, whenever you get a sec, noting that this is the start of pump 7, which will be 252.
He's about to hit that little rock, isn't he? Or is he going over it?
Is that a rattail?
Looks like.
I can tell you the volume filtered.
Stand by.
Take more zoom if you got it.
Let me just get my stuff together.
Nice tracking on that.
It looks like a giant tadpole.
Coming wide. A skeel.
Yep.
Looks very similar to the one we sampled on our first dive.
All right, I am ready for the mills for pump 5 whenever.
18970.
18970. Yep, got it. Thank you.
For those of you just joining. Oh, go ahead.
One sec, Matt. Sorry, that'll be 252, and it was like 2 minutes ago. NA pump number or sample number?
What's that?
NA174-252, which was the start of Multipuffer Pump 7. Thank you. Yes, pump 5 ended at the same time, although it actually may have ended sometime before, uncertain yet. Thank you. Go ahead, Matt. Thanks.
Yeah, no problem. Yeah, for those of you just joining us, my name is Matt Holden. I'm the Science Communication Fellow on the 12 to 4 watch. We just had our watch changeover, and so we are currently exploring a seamount south of Bakar Atoll This is a previously unexplored seamount. It was mapped earlier today by us, and it is located within the National Marine Sanctuary of Bikar and Bokak in the waters of the Republic of the Marshall Islands. Some of the objectives that we're going to be seeking on this dive are similar to previous dive objectives, to basically collect and observe different biological and geological specimens. Some of these biological specimens may be possible range extensions or even new species. I heard that the previous dive watch may have collected a new species, which is really exciting. And then our geological samples are going to tell us a little bit more about the history of how these environments were formed and the history of life on Earth. Super excited to be with you all. Please send us your questions.
Can you look down here?
Yeah. We'll also be collecting some eDNA or environmental DNA samples.
Oh, it's a zoantherian backpack and a hermit crab. Want to snap on that?
Yeah, you can.
Yeah.
Zoom in as I get closer.
Oh.
Yeah, it looks good. Yeah, you can zoom in.
Okay.
Whatever you need. Yeah.
Oh my gosh, that's great.
Oh, that's a big backpack.
Hermit crab.
He's a big backpack.
With a giant backpack.
The ratio of backpack to crab is...
Oh, actually, interesting. It's, yeah.
Oh.
We collected one of these a few years ago off Johnston Atoll. They look really weird when the backpack comes off. Really weird.
My students look different when they take their backpacks off too.
Hey Leela, have any Niskins been fired so far?
You got more zoom video?
None of the Niskins have been fired.
Take more zoom, we'll tilt you down, yeah.
Thanks.
Yeah.
This is actually pretty comical.
It really is.
It's moving so slowly.
You can't see me if I move slow enough. You think I'm just a zoantherian.
There was a Homolidae crab that was in the sediment last night. I don't know if anyone saw that.
Oh yeah, I remember. Mm-hmm.
That was really interesting, and interesting because that's deeper than their usual depth. Really neat to see that.
Yeah, this is wild. It's so funny.
Yeah, this is a good example of symbiosis.
Yeah. Could you tell us what benefits they're gaining?
Yeah, so I guess you could call it a mutualistic relationship. So the zoantherian is getting a really nice mobile home. And probably better feeding strategies than if you were sitting stationary on a rock. And then the crab is probably getting some sort of camouflage, either chemical or physical camouflage.
Could the zoantherian also like pick up little bits of whatever the crab is eating, or is it like
yeah, it probably could. Yeah, it probably could. Yeah, these crabs are pretty scavenger-y feeders.
Thanks, video. I'll probably keep going here.
Coming wide.
That's really cool. I guess we can go ahead and do a round of introductions. Again, my name is Matt Holden. I'm the Science Communication Fellow for the 12 to 4 watch. Hi. When I am not at sea, I am a high school science teacher in Fayetteville, Arkansas. Super excited to be here exploring with you all on this previously unexplored seamount. Steve, would you mind introducing yourself?
Yes. My name is Steve Oskovich. I'm the watch lead for the 12 to 4.
Yeah.
And I am one of the biology science co-leads on this expedition. I'm an invertebrate biologist. That is a biologist that focuses on studying invertebrates. Not a spineless biologist. Clarification.
I respect that.
It's 12 at night, you know.
Oh no, I hit a button and I don't know what I did.
So I come from the Smithsonian Institution's National Museum of Natural History, where I focus on genetics and genomics of deep sea animals, much like the ones that we're collecting across these dives, and trying to identify them using molecular genetic tools as well as understanding and identifying patterns in environmental DNA that we might collect across the landscape here.
Awesome. Super glad to have you on board, Steve. Super knowledgeable. I've learned so much from you on these dives. It's incredible. Mariana, would you mind introducing yourself?
Hello everyone, my name is Mariana Poznansky. I'm from the Marshall Islands and so grateful to be here.
Yeah, we're so happy to have you. Just having your cultural expertise on board has just been absolutely amazing, and so happy we've got to learn from you. Leela, would you introduce yourself?
Hey y'all, I am Leela Madhava. I'm the data logger for this shift. I'm originally from Chicago, but now I go to school at University of Washington, Seattle.
Very cool. Happy to have you on board, Leela.
I'm happy to be here.
All right, let's see who's next. Annette, would you mind telling us a little bit about yourself?
Sure. I'm Annette Govindarajan. I'm one of the science co-leads. I'm a biologist at the Woods Hole Oceanographic Institution. And I'm here collecting eDNA samples, environmental DNA, and trying out some new technology with autonomous samplers that we have, the Multipuffer on the Hercules.
Very cool. So excited to have that new technology on board. I was trying to remember yesterday when you told us how many liters we had filtered.
It's a lot.
Was it like 1.6 million or something wild
something like that? Yeah.
Absolutely incredible.
We're getting typically around 200,000 per
dive.
Sample.
Per sample. Wow.
I'm sorry, 200 liters per sample.
Okay, gotcha.
Yep.
Yeah, that's really, really cool. Dan, would you mind introducing yourself?
Yeah, absolutely. I'm Dan Kirilov. I'm our video engineer for this watch. This is my first cruise. I'm a little sad for this to be our last dive, but still loving it.
Yeah, I feel that pain, Dan. I'm also sad this is our last dive, but I guess we did get lucky. We get 3 watches on this dive, when every other watch only has 2. So pretty excited about that.
What is going on here?
What is that?
It's a sea cucumber.
Cucumber.
Yeah, you can see the gut.
Oh, yeah.
It looks heavily ballasted.
It is well anchored, as they say. Yeah.
That's wild looking.
Yeah, we've seen this for the past several hundred meters. I saw a couple before I went to sleep last night on the boarding watch. Yeah, a little bit. It was really therapeutic. Just a few hours, though. I don't want to get too much.
Yeah.
We can't get too crazy with it.
Yeah. Can't get too crazy. Don't get too much sleep. You might stay down there.
Yeah, sometimes when I can't fall asleep, I just count sea cucumbers. And just, that's what happens.
Is that the
What are cucumbers if not the sheep of the sea?
Right,
yeah. Well, in some sites they are, yeah.
Looks like there's some little tiny something stalked below it, too, also. White floating at the very bottom.
Tilt down a tad.
So, where are they looking?
Oh,
down here?
Down here, yeah. That looks like a bryozoan. Bryozoan, probably.
Oh, that's pretty small.
Thing. Oh, yeah, there's another bryozoan here. So, lots of bryozoans. Okay.
Next video.
Yep,
yeah,
coming
very
on.
cool.
Happy back here.
Looks like the res up to 5 psi. That's nice.
Yep. We did good filling that.
ROV pilots, would now be a good time to introduce yourself.
Yeah.
Cool. Go ahead, Rye.
My name is Ryder Wolf, and I am sitting on the co-pilot seat. In the co-pilot seat.
Awesome.
Perched atop it like a bird or a gargoyle.
Just perched up there. Love it. Awesome. Glad to have you on board, Rye, and be working with you. Levi, would you introduce yourself for us?
Yeah, my name's Levi Unema, and I'm flying Hercules right now. And I'm from Newport, Rhode Island. And this is also my first cruise this year. So this is our last dive of this cruise, so kind of bittersweet.
Definitely. Yeah.
But awesome to be here.
Yeah. I think that means since it's our last dive, we're going to see some really amazing creatures. At least that's what I'm putting out into the universe. And Gabby, would you mind introducing yourself?
Yes, I am Gabby Espino, originally from Kailua, Oahu in Hawaii, and I'm the navigator, and I'm also a C4 mapper during this expedition, and I'm just so happy to be part of this watch team.
Yeah, we're glad you're on it as well. It's been a great watch team. I've really enjoyed getting to know everyone a little bit better. I feel like we're in one big happy family. I love it. So again, yeah, we are ready for your questions. If you want to know anything, send it my way and we'll do our best to answer them all. Again, if you're watching, you're getting to see this live for the first time, just like us, so consider yourself an explorer as well. We actually have people from all around the world watching right now. I think
you want to do another 20 meters?
Copy that.
If you're ready for it, Copilot? Yeah, all right.
We've got some people in the U.S.,
okay.
United Kingdom, Australia, Germany, Canada, Brazil, South Africa, Portugal, Philippines, Marshall Islands, Guam, France, Spain, the Czech Republic, and Belgium. So all around the world, people are watching and exploring along with us, and we're happy to have you.
Whenever you say all around the world, I just think of that song.
Dude, same. Around the world, like Daft Punk,
yeah.
Around the world.
Great song, great band.
We have the Bluetooth, we can do it.
It's old school.
There's a really cool music video too.
Around the world.
I just got a comment from a 7th grade class in Brazil. Happy to have you all watching. You guys are all explorers along with us, and keep on learning and listen to your teachers.
Sounds like something a teacher would say.
That's true.
Bom dia, Brazil.
Awesome.
So the water on the lens. Oh, yeah, it's probably just some like goo from gelatinous creatures.
Just got a question. Maybe Steve, you can help me answer this. Is this a location where we would find a gulper eel?
Possibly.
Possibly.
Yeah,
they're more midwater.
They're midwater, but sometimes they can drift down. In fact, yeah, it's depending on
pictures of the gulper eels?
How shallow or deep we are,
They're
Hercules lights will attract and kind of carry down a lot of midwater animals.
They're crazy.
Okay. Yeah.
It's probably not
like
very common,
it's awesome.
but the lights definitely attract fishes. And if
Yeah.
it doesn't attract the gulper eels, it can attract the prey of them. But we've had some really great midwater fish situations videotaped because they're attracted to the lights of the vehicle.
Yeah.
Let's look like that.
What are y'all laughing about over there?
Oh, am I on SBL? Jeez,
Yeah.
sorry. That's my bad.
Hot mic. Hot mic.
That was on me. My bad. I thought I was muted on SBL. I'm not even good. We're just looking at photos of gulper eels and laughing.
Oh, I
have
love
no
it.
excuse.
Yeah, they're so crazy looking. We have fun here at the
Sorry, Steve.
12 to 4 a.m. watch. Yeah, as Steve mentioned, we've seen some really incredible fish on our dives. Super excited. Hopefully we'll see some more.
You see how there's a droplet of water? Is this on the outside or the inside of the dome, you think? Because it's been there the past couple dives.
To be honest, I'm not sure.
Yeah, okay. Minor detail.
Could be like gelatinous on the outside, or it could be a little drop of water on the inside.
Looks like there's a couple. Like, yeah, there's definitely a few more to the left and up. And
it's like once you see it, you can't not see it.
Yeah, it's one of those things, yeah.
We need AI to take those out for us.
There's a white something right there in the sediment. Can we take a peek at that when we're up there?
I'm going to zoom in partial video so I can try to find it as I land. I don't see it, Steve. Can you help me?
I don't see it either.
I think it's down a little bit. Yeah, it told me down.
I lost it.
I think it was right in that sand patch there.
Yeah, I was guessing it's a Xenophia 4.
Huh.
Okay. Never mind.
Okay.
All right, coming back wide.
Maybe some sponge rubble on the top left up there, maybe?
Copy.
I'll be right back, guys. I got to get a picture of the crew members about to do an interaction. For those of you listening, we do live ship-to-shore interactions, and we're about to have a marathon of them. We've got 8 today, so we're really excited about that.
All right.
No sleeping.
Should we keep the moves coming, Copilot, or do you want to wait a minute for the next one?
Yeah, I think we can do another one.
All right. I think 20 is fine. I think it's going to get steeper.
Yeah.
Oh, hello, fish. All right, video. Let's go in. Let's see what we can do together, Dan. I'll try to center you up. Go, go, go.
It's Cuskeel.
Oh, scared him.
The small ones are really jumpy.
Yeah. If you got more zoom, you can take it. Yeah.
Nice frame up on that. Nice.
Dragging my toes.
All right. I think that looks good.
Okay. Makes video.
Yeah.
All right, I'm back. I know you guys missed me.
You missed Cuskeel.
I thought you were about to say an octopus or something. I was
oh my
like,
god,
what?
can you imagine?
I would be so sad.
Dear octopus, 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 octopus.
And I'm sure the Cuskeel missed
caves.
you as well.
Oh, thank you. I think it did.
I feel like this is like a high stakes I Spy. You know, like we're all scanning these rocks for anything.
Right.
The octopuses will usually, they usually sit in two ways. They'll either sit on the seafloor in kind of a lump and look like a fleshy rock, or they'll kind of drift in from the side.
Oh, what's this gelatinous video? See white transparent bottom screen on our land.
Oh yeah, I see it.
And then you can come in.
Oh yeah.
Tunicate or something?
I think it's a tunicate, yeah.
Right.
Megalodicopia.
Megalodicopia?
Megalodicopia.
Megalodicopia.
It's one of the genus names for these predatory tunicates. So this kind of works the same way as the fly trap. You know, animal goes in, mouth closes, animal digested, rinse and repeat.
Oh, there's another of those little white branching guys in the lower left there. See that?
Yeah, the bryozoans, yeah. Probably one of the most abundant things at this site, these bryozoans.
It's really interesting how a lot of the things we see down here are transparent.
Yeah, yeah. Pigments are really expensive, energetically expensive to produce.
Yeah, that makes sense.
So if things can live a more lean energy balance in their bodies, they typically will aim for translucence and decrease weight.
Video. Thank you.
Less weight means less cells means less energy metabolism demand.
Really cool.
And the temperature here is only around 1 or 2 degrees, so their metabolism is directly related to the temperature of the surroundings.
Yeah, always an energetic trade-off. What kind of food would those tunicates eat?
Good question. I'm not sure if it's if they're preying on just particulate matter, organic matter, or if it's actually an animal, living animals that might be attracted. Sometimes, you know, this is something we don't really know well. Is maybe they're fishing with a type of bioluminescence or chemical signature to attract a certain prey. There's a white, maybe cup coral on the left side of that rock.
Good eye.
You want to do a snap drive-by, or are you going to wait?
Want to snap it?
Yep.
I just see staining.
I think it might be
Where
rubble.
Okay. I'm coming down, I'm coming down, I'm coming down. I don't see it, Steve.
Yeah. Okay.
It's so
Forget
hard.
I think it's just
coming out.
Yeah. We could probably do 20 more.
Optical illusion.
Yeah.
You ready for it, co-pilot?
Yep.
All right.
Kind of feel like I almost see it now that we're zoomed out, but
maybe the cup coral is the friends we made along the way.
You're all cup corals to me.
All right, yeah.
Rock City here.
This pace is working really well.
So we're now overlapping the lowest or deepest depth we've dove on all of our previous dives. So we have a continuous view of every depth range from 33.25 meters up to 950 meters on this expedition. So
wow,
we'll have a lot of data to interrogate after this cruise is over.
Yeah, for those of you watching, we are currently at 2,732 meters below the surface.
Oh, hello.
Who do we have here?
Cricogorgia.
Looks like someone might be home down at the bottom, or is that just the base, maybe?
Yeah, there may be a bunch of stuff in here. So this is a Cricogorgia colony. There might be some squat lobsters. And then the base always looks kind of free of sediment, and that's kind of neat because sometimes there can be
I'm gonna get you close.
other animals that feed in and around the base of the corals.
That.
The last watch did suction an entire Cricogorgia, which is impressive. It's a
Wow.
It's a bold move.
Whoa.
Whoa. Probably
All right,
not this big,
come in.
but
Sorry.
Anybody home?
Tilt me up a little bit.
Yeah. There you go. Sorry,
You're
that's
good.
disorienting nose.
so this Cricogorgia has at least a couple of squat lobsters, Europtychus species.
Bring the camera left a little bit.
Yeah.
When you get a second.
You want to see the stalk in the middle of it? There.
Yeah.
We call
that's a
this
great
type
zoom.
of Cricogorgia bottle brush because it's dense and tall, taller than it is wide.
Got more zoom, or is that it?
Oh,
That's
okay.
full.
Yeah, that makes
Like
sense.
a bottle brush, yeah.
Yeah. All the ones I have in my lab at school somehow disappear every year.
I'll zoom out a little
I don't
bit,
know
see if
how
we
they
see
keep
any associates in here.
growing legs and walking off.
And then we'll tilt you to them.
There's one right there, just to the left, looks like, right?
Oh yeah, one of
here.
those doobie daubers.
Oh yeah,
Doobie
actually,
daubers.
that's a
That's the
that's
technical
a
term.
that's a ctenophore.
Oh, wow, cool.
So you can see the ctenophore here, and then the
Yeah.
long lures it uses.
Oh, that's cool.
Yeah. Are there any more? Can we just scan the whole thing?
That's
So
really
come
cool.
wide a little bit.
Do you want to collect it?
Maybe.
I think
Let's-
it would be useful.
Wow, those
It's
are
kind
long.
of-
We
oh,
got a
yeah.
squat lobster up there.
Yeah.
Sorry, just give me a second.
It's a really big piece to have to grab, and they're really difficult to grab without getting the whole thing too. So if we can see if there's more, maybe we can target our collection in a
Do we
better
want
way.
to look at that big squat lobster at the top?
Sure. Tilt me up to
Yeah.
spot.
The big one, yeah.
There are a couple of more ctenophores a bit lower. Oh, and there's one right here too. I see some
A
tentacles.
little one,
Oops.
yeah.
There's one up top left too. See by the green laser dot?
Yeah, there's a
Yeah,
little
yeah,
one
I
there.
see it. Uh-huh.
Damn, when you get a
Oh,
stick,
yeah.
can you
Eel
give
in
me
the
Okay,
more
so
address
there's
on
a
how to
few
link
in
it?
here. Yeah. Yeah, I think we will go for a collection here. If we
Okay,
zoom
totally
out,
the
we
ship
can
back
see how
centimeters,
much
or
of this we actually need to
What do you
take.
think, Rye?
I think.
Because I think we got some swing left in us.
Yeah, we definitely do. Yeah, let's go back.
You want to go back
'cause
10 meters?
there's yellow
Someone
right
in our
the
chat is
center
very excited about
Just go
tunicates.
reverse.
So will
whatever
the
last move
target
was.
be the
10.
tunicates?
No,
Cool.
the target will be the coral, and the
Okay.
everything else is.
All right, Steve, what would you like to sample here?
All right, let's.
what's
Can we get the lasers on it to see how big this actually is?
Yeah. See the green one there on the left? There you go.
Yeah.
10 centimeters across.
Yeah, it's a really big. Okay, well, there's several ctenophores up on top, so I think it makes the most sense to go aim like right here.
Okay.
Under the
Perfect,
squat
thank you.
lobster.
Under
While
squat.
trying not to touch the squat lobster so it bails out. You know what I mean?
Roger.
So we want the squat lobster.
Yeah.
Yeah, right under the squat lobster.
And the ctenophores?
And the ctenophores. Yeah, there's several throughout the upper third of this. So, and then there's also, I think I saw a squat lobster down lower too. So there's another one there. But I think we're just gonna aim for like maybe 50
want it all.
centimeters.
Sorry about that. I'm gonna get the ROV right here.
Steve, before we finish all of our shifts, I really want to draw on that screen.
Oh yeah. Yeah.
Also had a really good comment about when you said interrogating the data. It made someone think that
Okay.
the data's been tied
Unparked.
up in a chair.
Yeah.
The scientist is demanding answers.
I really like that word.
Okay.
We often say interrogate the seafloor.
And this is going in a box.
As a euphemism for exploration.
Probably a box, yeah. So we'll cut it.
I like it too.
If Atalanta goes too far, after we cut, I'll just float it up.
And this is going to go in the forward mile box because we have a whole open compartment, right?
We have a coral in 240, but sorry, in lambda, but omega's open.
Omega, okay. What's the coral in? Oh, it's a bamboo. Okay, I saw it. All right, yeah. So we're going to go to omega.
There we go.
So just under that squat lobster there.
Am I going to be pretty far up near the brow bar?
Yeah, I think, yeah. I can...
Can you tilt me up just so I can make sure I'm not grabbing into it? Yeah.
Yeah, go below that squat lobster there, yeah.
Yeah, and I see. So we want like, oh, I see. Find it in space.
Nice.
Have I got those on the bottom? Yeah.
Yeah.
Nice, okay. Okay, and we want a little bit lower? Or right there?
That's good.
Yeah.
Okay.
Kind of want to try to avoid touching the squat lobster because if it it gets touched, it might bail.
It might bail. All right. I think that's below where he is.
Yeah.
Agree, Steve?
Yeah, I think we should go try that.
It's going to be a bit.
Wait, see how you go center of the jaws?
Yeah, I see.
Wrist left or shoulder left.
Yeah, look how many of these ctenophores there are.
Yeah.
Like that.
Yeah. That'll get it in the cutters better, yep.
This ctenophore is named Jophiella, which, if you don't like the way ctenophore is spelled, you're going to love the way Jophiella is spelled.
There we go.
Nice.
Just because these rocks are here, I'm going to drive the ROV and reset it more in your view. Cool? So I'm going to take off here.
I'm just going to freeze here.
Yeah, you hold it right there for just a moment.
Looks a little bit like Herc is flying around with its like lollipop or something.
Like cotton candy or something.
I put the spelling of the ctenophore associate in the sea
Wow,
log.
you weren't kidding.
Oh my god.
T for Jafia.
I'm sorry, I don't make the rules. That's just the way it's spelled.
C as in ctenophore.
Every time I write ctenophore now, I hear your voice saying that.
You want me to open the... I'll probably wait to open the box until we stop moving.
I think we need to do a Sesame Street song about, silent letters and
I'm not submitting the sample because it's not in the box yet.
Yeah,
that's fair.
Well, I figure we'll be within a few meters, so the oceanographic will be...
We'll just have to do might take a few more minutes to do the vid caps.
His is super cute, though.
I'm doing the vid caps, so that's okay.
With his
I'll forgive myself that one.
Yeah, right. So just make a note. There's probably
all right.
There's like at least 6.
You can tilt me
ctenophores.
down.
I'm going to retract the camera.
Yeah.
And it'll take some time because they're very small to pick them off, but we might want to isolate some of them when we process this sample.
Is there anything floaty in these boxes we got to be concerned about? Or?
There shouldn't be. On the left side, there's a bamboo coral, but it's a sinky.
Left side bamboo, all right. All right, I'm going to open your box.
That scientific terminology.
But you probably don't want to open it too fast.
I'm going to try to slow
down. You got
down.
a view on it on BioBox or Bubble as well. Can you, Does it matter where it goes, Steve?
Omega, please.
Omega. All right. All right. Oh, running away. All right. Here we go.
Forward box close.
Very nice.
Nice.
Wunderbar.
That's a really...
It looks like that.
That's
Lobster
pretty much a
stayed in there?
flawless collection.
Okay, perfect. I guess we got a little bit left in those jars. I can just let drop. All right, putting the jaws away.
Nice sample.
Very good. As always, our ROV pilots, they are pros at collecting these amazing organisms and rocks.
Oh, we got us egg, I think.
What's up?
Oh, just the arm.
Oh, saggage. Copy. Ooh, let's go for fish.
Eel?
I'm going for the chase video.
All right.
All right, Dan, let's do it.
Let's do it.
Together.
Our powers combined.
I'm going to try to stay this altitude. I'm racing over there, coming in hot.
Cool.
Ooh.
Yep. Keep the zoom.
Nice.
Good.
That's full zoom.
We're seeing this live.
Okay.
This is.
Oh, too far.
Is it a cuskeel?
The Nautilus.
Yeah, it's a cuskeel.
This is a cuskeel.
We're going to pirouette. We're going to come around
Dan
starboard.
and Levi's Cinematic Universe.
Movie magic.
Doing a dolly here. Trying to get the broadside. All right, go for eyeballs. Go.
Wow.
You're a natural director.
Okay. Okay, float it. Steady. Focus. Okay.
Turn to the camera, turn to the
kill
camera.
lasers for cinematic.
Beautiful. You're giving us space.
He's coming out of frame.
Here, I'm killing the laser. All right.
Eyeballs, go for
Sorry, I
focus
wasn't aware I was
eyeballs.
supposed to.
You're giving us eyes.
perfect.
Focus eyeballs.
That's a highlight. That's
Focus.
making the gallery.
Focus
Oh,
eyeballs.
yep. Look at the camera.
Focus eyeballs.
There we go.
So, yep. Ooh, bye-bye.
That was amazing.
That was killer.
End scene.
Now the hard part to find the music to go with that.
Don't just ignore the dust on the ground over there for my thrusters. My heart is beating so fast right now. Yeah, look at that. I don't even know what heading I am.
All worth it for the cuskeel.
Yeah. I think that was his debut movie too. I don't think he's ever acted before.
He's a natural.
Yeah.
Jinx, you owe me a soda.
All right,
You owe me a soda is not a small thing on this ship. Those have value.
I was going to say, you're going to be waiting.
I'm going to be waiting until port.
I finished all mine before the transit ended.
I was going to say do a move, but I'm a little bit behind, so I'll wait a minute. Yeah. All right, video, let's do the snap the anemone. And I'm almost on the ground, and we're landed.
Yeah, anemone.
Yeah, I'll tell you now.
Yeah.
Very cool.
I'm incapable of operating my own tilt.
It's okay, you got your hands on the sticks.
That is full.
All right, I have a request.
Yes.
Can we pick up that rock with the anemone on it?
Sure, let's do it.
Cool.
All right, zoom out.
Yep, coming wide.
It's not too big, which is nice, and we don't have a good identification. Anemones are really difficult to get species level IDs on because...
Which rock?
The one with the anemone on it.
I think it's loose.
I didn't hear that part. Honestly, I just heard rock and then I zoned out.
All right, stay with me, your eye.
That rock, don't you
So I think
see?
we're gonna
The
go
one
on
right
starboard in
there.
one of the outer compartments.
Looks different than
We
all
have
the rest.
rocks in all of the inner ones, but yeah, outer ones are empty.
Okay.
Try to get a better land spot.
Oh, that's really close.
Are we worried about them getting the anemone getting crushed?
No,
Yes.
it'll probably withdraw, but
Okay,
I mean,
cool.
we probably don't want to put a big rock on top of it, but we can put smaller rocks
Bonus
adjacent to it.
All right, you settled out?
Yeah, okay, go.
I
Can
can
you
bring
tilt me down?
anemones
so
are
sure.
super difficult to work on. In part because some anemones get so
pristine condition.
massively large that they're impractical
get
to
it this
collect
way.
because they can get so dense that they can't be properly preserved, and they have to
Maybe
inject
shoulder
preservative
left
into them.
more.
'cause they're
Shoulder left.
You inject what into them?
Like that?
Preservative.
and then wrist.
So
And
sometimes
then
they
wrist rotate which way?
when they
Wrist
tighten,
right.
they get so
Leg and
tight
shoulder
so the
left.
preservative
Yeah.
doesn't
Wrist
penetrate.
right.
And so
Gotcha.
sometimes you have
Leg.
to preserve them with a syringe,
Oh, like-
inject preservative
I
into them.
see.
Yeah,
Like this.
There you
Typically,
go.
And
I can move the vehicle too if it's not a good spot.
So
Yeah.
This, you don't have to answer this, but if I were to pick between the rock and the anemone as the target, I assume it would be the anemone.
Oh, it's moving. I think it's going to move.
The rock.
I think it's going to move.
I'm going to say the rock.
I think it
rock?
is.
Rock? Okay. Yeah. Just if I'm sorting this as like a rock or...
I would say it's a rock. It's a geo first.
I think I need a better view of this.
Anemone is an associate
otherwise
of the
I might
rock.
just end up squashing this.
Zoom out video.
Yep, copy. Coming wide.
Can you take the arm?
And also, anemones are really difficult to work with for eDNA reference libraries because most of them are formalin fixed. Because that's how we identified them. So formalin isn't great for DNA.
Right.
No thanks. Oh no, it's coming back.
Get out of here.
That's good.
So fresh material is better.
Always better.
Just the fingertips. The ROV is going to take out this poor anemone if I don't move the ROV real quick. Okay.
What are you trying to do?
I'm just going to land right here.
Yeah.
In a more steady position. Okay, so I would go just fingertips. Like just the jaw.
Onto the rock,
onto the anemone rock, yeah.
Yeah.
So the anemone might be like...
It'll be in the jaws.
Yes,
like,
protected in the jaws, I can zoom back in.
Yep.
So just go for fingertips.
I think I want to go like lengthwise though, like that. You know what I mean?
Beautiful. Just get the tips underneath it a little bit, but not too much. Yeah.
I think that's...
Oh, the rock's bigger than that?
It's longer.
See how the... Actually, you got to go deeper. Yes.
Go like that.
Yes.
Yep.
Yes.
Oh, nice.
Love it. Work it.
Nice.
Do I have it?
Gingerly, yes. You have it gingerly. Yep.
Gingerly.
Beautiful. It's just on the fingertips. Okay. Which box are we going for?
Starboard.
Okay.
Either of the...
Really?
Other one.
Can we go... do we have to? Okay.
Yes, we do.
Let's just go to the hard box. Okay, yeah.
It's the two big ones are open though, right?
All right. Cameras, starboard box.
You can tell it's
okay.
12:48 a.m.
I don't know if 12:48 a.m. justifies it. That's not that late. If it was like 3, that'd be one thing.
Got sample program?
It's great though. I'm loving it. Loving this camaraderie.
So we're going to go in E or F.
All right, rounder.
How can I help you? Are the lights on?
Yep, lights are on.
All right.
I think you can bring the box out. I think
Your wrist is pretty far. Oh, yeah, you're good. Let's see what's it's way up in the air. All right, he said E or F.
Copy.
Yeah.
Doesn't matter. Dealer's choice. Whatever you got.
Let's do E.
Okay, E sounds great.
I think I am right over E. But let's make sure.
Should be a sinker, I believe. If it's
I would hope so.
Thanks for that one. That's great.
Is there floaty rocks here that I don't know about?
Yeah, I'm going to try to place it in there so that it falls. Look at how considerate I'm being.
I couldn't help myself.
Amazing.
Rock and anemone collected.
Ride the wolf on the manipulator. Starboard box
closed.
closed.
I submitted it before the box was closed. I'm not supposed to do that.
Oh, yeah.
What if the rock flew out back into the box?
The anemone propels it.
What would I do then?
Well, sometimes these rocks get stuck in the jaws.
Standard operating procedure, gray area.
We'd have to wake up Michael and then it'd all be over.
All right, give me back my dive salvo.
Roger, dive custom.
So, so we're going to say this one's an Actinostolid. Actinostolidae family. None of these have species names on them.
Cool.
none of them from the Anemone
good.
Oh, wow.
So
yeah, not a lot known?
Not a lot known.
We did it. Nice job, Ryan.
Thanks.
Good job.
Both of y'all did a
Pop
great
off,
job.
ROV.
Rock a clock.
Rock
Should we do a 20-meter move, right?
Featuring anemone. Yeah.
Yeah, let's do that
I think
20-meter move,
that was its
305
first
or
film
whatever.
as well.
Right.
Whatever gets us waypoint 7.
Which bearing looks good to go up the hill?
So I'm
we're kind of cutting it right now. She's like, see how it's pretty far off to Atlantis Port? That's
I'm currently
uphill.
340.
Oh,
so
340.
340.
You turn a little bit. Okay.
Yeah, I did to keep you in view.
Got it. Let's go back port then a little bit.
Yeah.
Which is the bearing to waypoint 7 there?
That's going to be 300.
Yeah, okay. That would put us.
Great. I like that better.
All right, we'll do bearing 300.
Sweet. She's looking 3.
Yeah.
So do you think ctenophore scientists just have a real love for doing a really weird letter for the first name of a species?
Yeah.
These ctenophores we collected are Jalfiella, starting with a T.
Yeah. Yeah, that's where the ctenophore T, it actually just went to that genus.
And then the J will go over to the next one, which will be like Astereotinidae, starting with the J.
Starting with the J, yeah.
Wait, Astereotinidae?
I just made that up. That is an entirely made up species name.
That's a pretty good made up species name.
Yeah.
Thank you. Thank you very much.
I think this is like a game, like whoever's naming these is like, you know what, I'm just going to make these all start with the wrong letter.
See, that was exactly what I was getting at. This was
like, that's
exactly
like somebody's like...
if you want to ask these questions, I think, so there's a conference series for ctenophore enthusiasts called Tinapalooza.
What?
Oh my God, that's the best
Is this
thing you could have
serious?
said.
I'm not kidding.
Oh my
That's
God,
fantastic.
I want to go.
Googling this immediately.
Where is it?
It's got to be Chicago, right? Like,
Yeah.
like, come on.
I think the last one was
That's
in
where
Florida.
Lollapalooza was, right?
Yeah, St. Augustine, Florida, 2016.
Am I
but
spelling Palooza wrong somehow?
Are you spelling it with a T at the start?
Not unless I check.
Remember.
No, I am spelling this
C
right.
as
I'm a
in
good
ctenophore.
Chicagoan.
As in ctenophore.
I think we got a little rock formation up here.
Wait, how do you spell Cnidarian?
With a C.
With a
Oh
C.
my God.
Is it really with a C? Oh, I thought it would be like
I
a
knew
K.
But the thing is that the character, the morphological feature, let me zoom here. The morphological feature that defines the cnidarians is called a nematocyst, and that starts with an N. So
A nematocyst?
Nematocyst.
All right, video. Let's.
Is that
let's take a
brittle
look at
star?
these
Brittle
little
star?
fingers coming over the dark
But
rock
the actual
here.
stinging cell is called a cnidocyte,
Cnidocyte.
which starts with a C. So
Of course it does.
yeah,
That's a little creepy looking,
it's the
isn't it?
backside of a bursingid, I think.
It's like
No,
Yeah.
like a hand going over
Like a
that.
horror
not a
movie.
bursingid, it's
Yeah.
an
Got
ophiocanthid
enough spikes for a horror movie.
brittle star.
He doesn't
It's
look
just
very
hanging out on
friendly,
the rock,
does he?
though.
That many spikes.
Okay, that's happy.
Okay, coming out.
Are they actually like hard spiky?
They are spiny, yeah. They're
Oh.
quite spiny. Yep.
You're not to touch them, Rye.
Okay.
Don't try and eat them.
Okay.
Okay?
Yeah, no, those
No promises.
those do hurt.
You're not going to eat them either.
It's not pleasant.
Forbidden spaghetti.
Yeah.
These web interactions are showing up on my screen, and they're showing a Chaunacops right now, and I just looked over, and like, I was like, oh my God, it's a Chaunacops.
We all silently zoomed in on a Chaunacops.
What is this? Is this a brittle star wrapped around an old dead sponge? All right,
Something.
You
Let
can
me do a
zoom
little
in.
snap.
Yeah,
All right.
Yeah, snap it. Let's see what we got.
I think a couple of them.
Oh yeah.
I'm going to pirouette it, I guess. Let's try to get to the other side
Actually,
of it.
I'm more curious about what these are.
Oh.
You
Okay.
see those gelatinous ball-shaped things there?
Roger.
Oh yeah.
Balls on dead sponge.
Yeah, what are the...
Is it just part of the sponge that's still
ctenophores?
alive, maybe?
Those are actually ctenophores, or not
Oh.
ctenophores, tunicates,
Oh.
Tunicates.
which actually doesn't start with a
That
C,
one does
but-
start with a T. I know that one.
Tunicate.
tunicate, yeah. Yeah, so that's a filter-feeding animal.
Do you need a closer, longer
you can
look?
see the inside
you can see
the little star.
intake and out excurrent siphons.
Yeah.
Oh, the tiniest star.
You see the tiny star.
Where?
Just right
On
above
there.
the tunicate in the dead sponge.
Oh, it's so
Oh,
tiny.
it's so small.
Oh my gosh, that is the smallest star I've ever seen.
It's adorable.
My
It's like a grain of sand.
Can we zoom?
No, we're full.
Ah.
My eyes are not good enough for this.
Third hole from the bottom.
Teleprompt.
this
We got to
is-
do
this
the
is where the
circle.
star
Yeah,
is.
circle.
Oh,
There we
okay,
go.
I do see it. I do see it.
So this is a tunicate. It's a chordate animal, so it's actually our closest living relative down here, next, aside from the fishes.
All right. I think we're good, Dan.
Cool.
This is
Coming
our
along.
closest
good.
living relative down here?
Aside from the fishes, yeah.
Wow.
They're actually, they're chordates, so
Oh, yeah. Okay.
They have a notochord,
Yeah.
which is dissolves after it settles, and otherwise they're
basket star.
That's crazy.
they're very
That
close
is
to
not
us
something that looks like it would
evolutionarily.
have a notochord.
Yeah.
That's
in the
so cool.
in the larval
I'm
stage they
extending your
do.
camera.
Yeah.
Okay, so only in the
Another
larval
Agree
stage.
well.
Yeah,
They're
and then
short
it's
of
Like we're driving a rock
body.
tumbler.
Rumble
That's
tumbles.
wild. That is so interesting. I can't wait to use that in my evolution unit.
That one's big.
It's so weird that it would be more rigid extended.
Yeah. It's gotta just be that.
Is this a Brisingid? I'm doubting myself now.
Love the coloration.
All right, I'm gonna land here, and we'll zoom in from here. I can't really
Do we
look
have
at it.
anyone on shore that is
All right, tilt me down a little bit,
requesting
and then Dan, you come in. Yeah.
Yep. No, not specifically, but
Go
they're
for center disc spines.
Just getting
they're
a
not-
lot of comments
yeah.
about tunicate.
Is it like
Yeah.
have like a proud bulb to it, or is it just
Looks
a
like
leg?
it.
I'd like 10.
Huh. I like the little balls on the end of their spines.
Yeah.
They're like tube feet or something.
No, the tube feet are actually on the underside of the arms.
Yeah, there they are.
Yeah, you can actually see the tube feet here.
Yeah. Got more zoom, or is that it?
Get
Oh,
more.
there it is.
There it
Nice.
is. That's pretty close.
So the interesting thing about this group is that most, originally they were described from waters around Scandinavia back in the 1800s. So a lot of families and genus and species names for these comes from Norse mythology.
Oh, cool.
Yeah.
So is this like Thor?
No, but so the family name Brisingidae.
You want to zoom out a little bit, Dan, and get a full full specimen maybe, and then we'll go. There you go.
Pagan folk, man.
Yeah.
Huh.
Brisingamen.
That's cool.
It's the
legendary necklace belonging.
The necklace of the goddess Freya.
Freya. Okay, cool.
Yeah. And there's actually a genus named Freyella.
Alright, I just started
really
getting yellow in my innermost
very nice, thanks.
sonar ring.
Roger.
Okay, coming out.
And we're off. Back up the hill. Time to go. Onward and upward.
Yeah, till you up. Did it just get like a lot steeper? I guess so.
Yeah, we should be coming upon pretty steep terrain soon.
Okay, maybe we'll do 10-meter moves then next.
Copy.
Yeah, judging by the Atalanta sonar, you can't see over it. It's right at the 20 ring, it's there, and then at the 40 ring, it can't see over. And it's hard over there too, which is nice. Copy, Dan, thanks. Yeah, you want to do 10 more meters at 300?
Will do.
Take a peek.
Come on, octopus. Show yourself. Reveal yourself.
They have pretty good camouflage.
Yeah, they do.
Although down here they normally are just kind of purpley or white.
Yeah, I wonder if that's because a lot of these organisms have adapted to not, see in the visible spectrum, at least that we see them. Maybe. I don't know. Theory.
I'd stir some up. Yeah. That's me. Should we get lasers back on?
Yeah.
I'm going to go for the white stock sponge here next because I'm waiting for some tether. We got a ship move underway.
I'm going to do a log, but I'm listening.
All right, video, let's come in on the white stock sponge. I'm going to land here, and I'll touch it down. Yeah. It's hard to get definition on those, isn't it?
Yeah, with the sand as well.
Saccocalyx glass sponge. We collected one of these a few dives ago.
Just had a question. Is this the last televised dive of the trip? So this is our last dive of expedition
Come back up.
NA174 before we get back to port on the 17th in Majuro. I do not believe that our next expedition will be doing any ROV dives.
Copy.
So much stuff moving around.
So we'll definitely make this dive worth it.
Yeah, we will.
All right, there's something white in the, oh, no, that's on my lens. I think we do another 10 meters. What do you think, right?
Just 10?
Yeah.
I guess, yeah.
Copy.
Just because it's pretty hard in that sonar.
Bridge nav.
Think we're gonna really see over it, like at this heading, or do we wanna?
What's up?
Do we wanna turn more into it, or?
We could.
Like.
After the next one, just because it's steeper.
Yeah, because it's. Yeah.
So yeah, we'll do this 10, and then we can turn port a little bit more into it. I like that.
For our audience members just tuning in, we are currently